Hello students. In this video lecture, we are going to discuss another interesting topic from your second chapter of 12th biology, and that is about the embryo sac of the angiosperm. Alright, so as you can see on your screen, the cross section of the embryo sac being represented. Alright, so on the frontal portion, which you can see the various types of cells being found. Alright, and we will be discussing about all the components of the embryo sac, what is its important, where is it found in this video lecture itself. Fine. Now, embryo sac is one such important, you can say, chambers of the ovule or the structures of the ovule that plays a very important role in case of the fertilization. Alright, so first of all, we will label all the components of the embryo sac. We will see that how the different components or how the different cells are arranged in the embryo sac, and then we will discuss about the fertilization process further. Now, if we talk about a typical angiospermic embryo sac, we are going to take up the angiospermic embryo sac. That means the embryo sac of the flowering plants. Alright, now embryo sac, if we have to define the structure, then this embryo sac is also known as female gametophyte. So this is the female gametophyte that arises from the single functional megaspore. And we have already seen in the process of the megasporogenesis that how the single functional megaspore is responsible for giving rise to the embryo sac. Alright, so that's why this embryo sac is also known as female gametophyte. Now, when we consider the embryo sac of the angiosperm, then the typical angiospermic embryo sac is usually 7-celled and 8 nucleate. That means 7 cells will be there, and inside those 7 cells a total of 8 nucleus will be found. So we will be seeing this part as well in the model, that where are these 7 cells localized and where are the 8 cells also being present. Alright, so this was about the definition of the embryo sac. Now let's move on to the labeling component or the labeling part of the embryo sac. So this is a typical embryo sac. Now typical embryo sac is having 2 ends. Alright, and this end is called as the chalagial end, and this end is called as the micropylar end. So embryo sac is having 2 ends, chalagial end and the micropylar end. And since this embryo sac is found inside the ovule, so ovule is also having the chalagial and micropylar end. So according to the position and location of the ovule's chalagial and micropylar end, the embryo sac has also been given the chalaja pole and the micropylar end or pole. Now, so this is the chalagial end and this is the micropylar end. Now, towards the chalagial end, you can see that 3 cells are being present. Alright, 3 cells are being found, and that is called as antipodal cells. And these cells are 3 in number. So you can see if I just label it out. So this is the first, this is the second, and this is the third cell, which you can see. Towards the center, you can see 2 cells are being present, and these are actually called as secondary nucleus, or you can say the central cell. If we consider it as cell, then this is also called as central cell. And this central cell is also called as polar nuclei. But once this polar nuclei they are fused, they are also called as secondary nucleus. So basically the central cell has been given 2 other names, that means the polar nuclei or the secondary nucleus after fusion that we are talking about. This we will be discussing in the double fertilization part. And you can see that there are basically the 2 cells being present. Alright, so this is the first one and this one is the second one. So if we consider the secondary nucleus, so 2 nucleus being present. But if we are going to consider all together this as a cell, so it's a single cell. Alright, so we can consider this central cell as a single cell, one cell. So in a single cell, 2 nucleus is being found. This is the peculiar feature. Now this cell which you can see over here towards the micropylar end, now towards the micropylar end there are 2 types of cells being present. One is the egg cell. This is also 1 in number. Alright, and these 2 cells which you can see over here, these are the Synergies, which are also 2 in number. So if we are going to count the total number of cells being present in the entire embryo sac and the total number of nucleus being present, so you can just count it out that the antipodal cells here is containing 3 cells. Here the central cell is the 1 cell, that means 4. Then egg cell, which is 1, counting it a total of 5. Alright, and Synergies having 2 cells, so making it a total of 7 cells. So that's why 7 celled. Alright, and if you are going to consider the nucleus part, so this is the first nucleus, second, third nucleus, fourth and fifth nucleus, then this is the 6th nucleus, this is the 7th one, and this is the 8th one. So making it a total of 8 nucleus present in 7 cells. So this we are counting it as 1 cell only, which is the central cell. Alright, so central cell is a single cell, but the nucleus that it is having is 2 nucleus. That's why. So that's why a typical angiospermic embryo sac is 7 celled and 8 nucleate, having 8 nucleus condition. Alright, so it is having 7 cells and 8 nucleate condition. So this is about the structure of the embryo sac that we have seen. Now, embryo sac is very, very important for the process of double fertilization. Now this embryo sac is a structure where the process of double fertilization takes place in case of the flowering plants. So we will also be talking about that process as well. Double fertilization in embryo sac. Now, since the word double fertilization is suggesting that there will be 2 types of fusions being involved. So a process that involves 2 types of fusions. So the first one is called as the Syngamy. That means the first type of fusion is called Syngamy, and the second type of fusion is called as the triple fusion. Now we will see that where we will see the exact location or the exact cell in the embryo sac where these 2 types of fusions are actually being found. Now we will first talk about the Syngamy. Syngamy is the process of fusion of the one male gamete, or you can say the first male gamete. See, we have already seen in the video of the pollen grains that the generative cell of the pollen grain produces 2 male gametes. This we know this fact. Alright. Now, since it is going to produce 2 male gametes, so the 2 male gametes are individually going to fuse with different cells. That's why it results into double fertilization. Alright. So Syngamy is basically a process of fusion of one male gamete with the egg cell to form zygote. Now see, we know this fact very well that this is the egg cell. Now the male gamete that I am talking about over here is going to come with the help of the pollen tube inside this one, like this. So the pollen tube is actually going to carry the 2 male gametes. Alright, these are the male gametes. In these 2 male gametes, one of the male gamete will be released in the chamber of the synergid and it is going to fuse with the nucleus of the egg cell. So this male gamete is also haploid and this egg cell is also having a haploid nucleus. So when they will be fused, they will form a diploid zygote. So this is about the syngamy. Now what about the triple fusion? Since the first male gamete has already been utilized, the second process, which is called as the triple fusion, this fusion involves the usage of the second male gamete. Now the second male gamete is going to fuse with this central cell. That means it is going to fuse with this secondary nucleus. Alright, we know that these are secondary nucleus. And the second male gamete is going to fuse with this nucleus only. So, a process in which the second male gamete, and this is also haploid. This second male gamete is also haploid. Second male gamete fuses with secondary nucleus. The one thing which is important to note over here is that you can see this point very clearly, that the secondary nucleus is already diploid. The secondary nucleus is here, is already diploid. Why? Because it is already having two nucleus inside them. Inside one cell is being present. So since it is already diploid, so this is 2n. So when the second male gamete is going to fuse with this secondary nucleus, this actually results into the formation of a triploid cell, which is called as triploid primary endosperm nucleus. So it fuses with the secondary nucleus to form triploid. That means 3n. Triploid primary endosperm nucleus. This forms triploid primary endosperm nucleus, which later on is actually going to differentiate into a cell. That means triploid primary endosperm cell. So initially the pen is formed. Shortly it is called as pen, P-E-N, primary endosperm nucleus. But this nucleus is ultimately going to undergo the process of cellular division and it will form the primary endosperm cell. So this is about the two processes or the two types of fusions involved in the double fertilization in case of the embryo sac. Alright, so we have seen that there is syngamy and then there is the triple fusion. So in case of syngamy, the male gamete, the first male gamete, which you can see over here, is going to fuse with the egg cell over here, while the second male gamete over here is going to fuse with the secondary nucleus or the central cell, resulting into formation of pen. So as I already told you that this pen, that means primary endosperm nucleus, is going to differentiate into, or it is going to later on form the primary endosperm cell. So pen later on forms the primary endosperm cell, which is triploid. This is all about the double fertilization process. But if we are going to see about the fate of the cells that are actually involved in case of the double fertilization, what is actually the result of the cells, or what is actually the product of the cells that are actually involved in case of the double fertilization. So fate of cells involved in double fertilization. Now, if you're going to look into the result of these cells, or the results of these cells, we can say, then the zygote which has formed as a result of syngamy, so that zygote which is diploid, that develops into embryo. So the zygote is going to develop into, ultimately into embryo. Primary endosperm cell which has formed as a result of the triple fusion, that develops into endosperm. So PEC develops into endosperm. So this is triploid and this is diploid. Embryo is diploid and endosperm is triploid. And what about the antipodal cells? This was about the fate of the egg cell and this was about the fate of the secondary nucleus. But what about the cells that are located on the upper and the lower side? That means at the chalagial pole and the micropylar pole. So the antipodal cells and synergids, they, once the double fertilization process gets over, these two cells, they actually degenerate. So this was about the structure of the embryo sac, and along with that we have also seen the process of the very important double fertilization that takes place in the embryo sac. Now, one thing which is left to be noted is that you can see over here that these are the synergid cells that we have discussed. Alright, these are the synergids. Alright, now these synergids, in the synergids you can see here some finger-like projections here, and these finger-like projections are called filiform apparatus. So these are finger-like projections, and the function of these finger-like projections, that means filiform apparatus, is that it will actually guide the entry of the pollen tube inside the synergids, so that the gametic contents, the male gametic contents, that means the two male gametes, they can be easily released inside the synergids. So that helps in guiding the entry of pollen tube inside synergids. And so they are going to discharge the male gametic content inside the synergids, and the process of fertilization will be rather convenient. Alright, so this was all about the structure of the embryo sac and double fertilization. Thank you.